Thank you for coming to today's session. I'm from Nature Technology Corporation, and we're the developers of a nanoplasmid platform, which is a vector platform which improves the performance of a wide range of gene and cell therapy vectors. And I'll give you a talk about that. We were recently acquired by Aldevron this year, and now we're combining our vectors and front-end work with their large manufacturing capabilities to provide a more start-to-finish solution. First, a little bit about Aldevron. Aldevron is a key manufacturer supplying critical raw materials into the gene and cell therapy space. So Aldevron is well known for manufacturing plasmid DNA, and you can see it has a tremendous capacity for making research grade, but also making a lot of GMP grade materials to support cell and gene therapy. The other thing with Aldevron, though, is a little different, is Aldevron also makes messenger RNA and proteins, and as such, can manufacture the central dogma of molecular biology. And that allows Aldevron to really service more than just plasmid DNA needs for clients that are looking for full solutions. So a good example would be in the area of gene editing vectors. So if you want to do a knock-in with a car, you're going to need a HDR template. So Aldevron can make you GMP HDR template, but can also provide you the Cas9 nuclease and a number of different options, very next-gen, like the SPY5 Cas9 nuclease that you can use for your gene editing. In the area of messenger RNA therapies, as you would all know, you make a plasmid first, you linearize it, then you do in vitro transcription, and then you do purify the messenger RNA. So Aldevron can do all that. They can manufacture the plasmid, linearize it, they manufacture enzymes for in vitro transcription, and can make messenger RNA. So Aldevron provides a lot of these different parts and pieces. Aldevron's also part of the Danaher family of companies. So for other things that you're not seeing here, one of our partners can provide that. So Aldevron can really be part of a full service supplier, CDMO, for all your needs and hook you up with other parts of Danaher for other things that might not be on this. Now, as I said, Nature Technology developed a nanoplasmid platform that improves vectors, gene therapy and cell therapy vectors generically. And so if you look at all this, every one of these classes has vectors in it. And when I talk about a platform to improve vectors, I'm talking about, for example, with cell therapy, you might be using a lentiviral vector. Well, you would manufacture the lentivirus using 4 vectors, typically. Nanoplasmids can be used for that. In gene therapy, AAV is typically made using 3 plasmid vectors. Again, we have a lot of nanoplasmid clients using our nanoplasmid backbones in place of the AAV. Same is true of any of these particular classes of vectors. So as I indicated, we were acquired for this nanoplasmid platform earlier this year, and from our point of view, we were having a thing where people wanted us to be a full-service supplier. We could provide the vectors, we'd provide research-grade DNA, but then they'd be like, we'd have to transfer them to a CMO. So we're very happy now to be part of Aldevron, so it's a seamless transition to GMP manufacturing. So what is the nanoplasmid? How does it generically improve vectors? The thing that is basically a very common truth of the industry is virtually everyone working on gene or cell therapy focuses entirely on their payload region of their vector. They don't really look at the backbone. It's, if you're an AAV scientist, you look at the ITR to ITR. You work on the promoter, the transgene. You don't look at the backbone. And the backbone basically is what provides replication and selection in the bacteria. And so typically what we see in these vectors is you have a puck origin. The puck replication origin is a temperature-sensitive origin that was developed in the 1980s. And it allows you to have a low copy of plasmid at 30 degrees, and then you increase the copy number at 37 to 42. So virtually all GMP manufacturing processes involve this temperature shift process where you have a low copy plasmid through the product growth cycle and then increase the copy number later. The other marker is like AMP or kanamycin. So as I said, virtually all of you have these old backbones. And one way you could do it is just ask your molecular biologist, take the backbone region of your plasmid and blast it, and you'll find something from the 1980s that it matches up with. It's just virtually all old. And so we got into making the nanoplasmids kind of indirectly. The first off was regulatory concerns about antibiotic markers. They did not want markers in the vectors. There's a number of reasons for this, but one, first and foremost, the marker gets transferred to the patient. You can get an immune response against it. You can also transfer antibiotic resistance. And so we developed a system called RNA out to get rid of the marker out of the vector. And we understood the regulatory guidance really wasn't just about antibiotics, it's about any kind of a protein. So we developed a marker that has just an RNA. It's a bacterial expressed RNA, it has no coding capacity, no homology to the human genome. It's only about 150 bases. It makes a 70 base pair RNA. And of course, for all these systems, we have an engineered bacterial host. In this case, the bacterial host expresses a Levan sucrase, which just is a sucrose concatemerizing enzyme. But the target of this RNA, the target of this RNA is in the leader of that. So the RNA, when you have RNA out of vector, you prevent the translation of that Levan sucrase, which means you're not concatemerizing sugar in the periplasmic space of the bacteria. So it's a sucrose selection. And all the cells that don't have the RNA out die in the presence of sucrose. So a very simple system. Where we got into making small vector backbones, though, was based on work where we were trying to get around gene silencing in the liver. So work by Dr. Kay's lab at Stanford had shown that if you have a large vector backbone and you deliver that to the liver, you get gene silencing. And their solution to that was minicircles. The problem with minicircles are a great research tool. In fact, there's a tremendous amount of data over the last 10 years using minicircles, showing advantages of small backbone vectors. But the problem with minicircles is they're difficult to translate clinically because they're so difficult to manufacture. You have to do an in vivo recombination. So we were looking for a solution that didn't involve the manufacturing problems of minicircles. So we took that puck origin, the temperature-sensitive puck origin out, and replaced it with a very small replication origin called the R6K origin. Now this origin also added another advantage. It has, the puck origin doesn't need, basically you put the puck origin in and it's autonomous. So you put it into any gram-negative like Salmonella or E. coli, it'll replicate. It doesn't need any transacting factor to allow it to replicate. The R6K origin has to have a rep protein. And so we engineered the chromosome to produce that rep protein also in a heat-inducible manner, so we could use the same manufacturing processes that people use for puck plasmid. But the advantages of that, of course, is that whereas the puck origin goes to a set copy number dictated by how strong the replication primer transcription is, we can drive as much rep protein expression as we want. So we actually have improved yields. So we ended up with this backbone with less than 500 bases, no coding capacity, no homology to the human genome, and we got the expected attributes, right? Eliminating antibiotics, increased yield because we can control the copy number, reduced transgene silencing. These things were also performed like that. The unexpected attributes was very high transgene expression. We expected it to be about the same as minicircles because minicircles, after recombination, have about 100 base pair backbone. But the expression was much higher than minicircles or plasmids. We hypothesized that the reason for that is the fact that it's hard to make minicircles, it's hard to get the supercoiling, so we think it's more of a quality issue. The other attribute was reduced transfection toxicity. So when you put a plasmid into a cell, you get a lot of innate immune activation, and the cell no longer, the cell can be killed by that activity. With the nanoplasmid and minicircles, you have much less transfection toxicity. So those two things, the high transgene expression and the reduced transfection toxicity, along with the regulatory improvements you get from eliminating the markers and coding capacity in the backbone, are really what gives you the improvement across the board with all these different vectors. And this is just one more safety feature that we can check off. For those of you who are familiar with the puck vector, because it can replicate in any gram-negative organism it gets into, there's lots of reports in the literature about concerns where a manufacturing facility has not had good containment and they find like the car or whatever it was that they were making in organisms downstream of the plant, also replicating in patient like cells if you have gut delivery and stuff. The nanoplasm, because it has to have that R6K or replication protein from the chromosome, can only replicate within the specialized host. So we feel like we've managed to combine the best attribute of plasmid and minicircle vectors with the nanoplasmid platform. We got the high yield and scalable manufacture of plasmid. We have gotten around the problem of that promiscuous puck origin to restrict it. We've dealt with a lot of the regulatory concerns and got rid of the antibiotics, captured the transfection, reduced transfection toxicity and reduced transgene silencing, and then also got the unexpected high transgene expression. And when I say high transgene expression, there's a number of scientific papers out there using the nanoplasmid. This is one of them by a group at Old Dominion, and they were using an optimized traditional plasmid, the GWiz vector, and then the nanoplasmid equivalent of that, and they were doing in vivo delivery to mice. And I just show this to show you that there can be pretty dramatic improved expression using the nanoplasm when you switch from a plasmid vector. So I'm just going to go over very briefly a couple of advantages that you may get with this. And one example is transposon gene editing vectors. There's been a lot of work published using nanoplasmids in this space, and what they find is that you reduce, just to back up just for a second, the therapies I'm thinking about, those cell therapies, is where you take the cells out of the patient, you nuclear affect the DNA into the cells, and then you integrate into the genome, either using a transposon or the activity of Cas9. And then you have to reach this viability threshold that the FDA dictates before you can put the cells back into the patient. And so you can see where the decrease in activation and decreased innate activation are two things that nanoplasm really helps with. So the cells are not knocked down viability-wise nearly as much as with a plasmid. So you have a shorter manufacturing timeline to get back into the patient. Work published by Genentech showed 2 times as many actual edited cells with a nanoplasmid versus a plasmid, 3 times as much as with a linear vector. And Poseida also published a lot of work using transposon, showing dramatically improved performance. If you think about it, you haven't knocked down the viability of the cell as much, so it's not surprising that the cells actually have better killing function, right? Because they're more viable, and that's what Poseida showed. And then in the area of poly-A vectors and viral vectors, the viral vectors are an interesting case, right? Because when you retrofit those 4 lentiviral vectors and the 3 AAV vectors, the virus you make is going to be exactly the same as the virus you gave us. So where would be the improvement? Well, it depends on what you're dealing with. The biggest one with the lenti is that the lenti are formed by budding off a cell. You have to have all 4 vectors get into the cell. They have to not be silenced or inactivated. And so we end up getting much higher titers with that because, you know, I think it makes sense in the sense that any cell that doesn't get all 4 plasmids and express them to levels that are functional won't produce the virus. And so that's the big one with lenti. AAV, the biggest thing with AAV is that recently it's been shown that AAV reverse packages the backbone in about 1% to 2% of all the viruses that go into the patient. So of course, people want to get away from having that marker transfer to the patient. And then another big one I'm not going to get into in great detail, but with AAV and messenger RNA vectors, we've switched the replication origin out of this vector. The nanoplasmid at R6K, the other vectors in the ART have puck. AAV and messenger RNA have structured DNA. A poly-A repeat in one case, a very difficult to replicate palindrome in the other case. The R6K origin does a better job of replicating through those without making little deletions or microdeletions. And when you combine nanoplasms with our engineered host strain Reviver, which is designed to stabilize those kind of repeat vectors, you can get higher yields of messenger RNA and AAV vectors and better stability of a poly-A and the ITR. So just back to the beginning, I mean, we've been providing this part for years to our partners and always had to pass off to a CMO for them to do their GMP, which is really not ideal. No one ever comes to us wanting to say, here, you give us part of the solution, we'll go find the rest of it. So I think we're very excited about being able to now seamlessly transfer off our GMP manufacturing to Aldevron, so you have one partner, and as I said, because we're part of the Danaher Corporation, we can help with other parts and pieces as well. So I'll stop here. I will be at the Aldevron booth after the session if anyone wants to ask me any questions.